Hi, Miss Nikki here. Welcome to Chapter 15. We're going to talk about the ANS, or the Autonomic Nervous System. And the very first slide will kind of do a recap of where we are from Chapter 12. Maybe you remember this figure from Chapter 12. We talked about sensory input and we talked about motor output. Specifically, we talked about somatic sensory. So this is messages from receptors, from special senses, or from the skin. We also have visceral sensory. So this is going to be from organs, right? Blood vessels, internal organs. And then we have output. We have somatic motor output. This is basically going to skeletal muscles, and we talked all about this in our skeletal chapters. This is what we're focusing on for Chapter 15. We're looking at the motor output that talks to cardiac muscle, smooth muscle, and glands. The autonomic nervous system is a motor division. So I know I stressed that on the last slide. I'm just saying it again. We're talking about signals being sent from the brain to the heart, smooth muscle, and glands. The sensory information that triggers the increase or decrease of this motor command comes from the visceral sensory inputs. And this is how we maintain homeostasis. I'm using this image from another textbook because I think it does a better job of explaining than the table that's in your textbook. But here, if we want to look at the difference between somatic nervous system, which is what we've been looking at this whole time, right? Up until now, we've focused on one myelinated neuron in most cases, releasing acetylcholine, affecting skeletal muscles, and the effect is stimulatory, right? So contraction would be a stimulatory event. When we look at the autonomic nervous system, we're going to have two divisions that we'll talk more about in just a second, but you can see that there's usually two neurons. Glands are a little tricky. Glands, there's only one neuron because the glands are actually releasing the hormone into the bloodstream. So that's the effect. We can see that in the sympathetic, it's going to release norepinephrine, and in the parasympathetic, it's going to release acetylcholine. So we're seeing a new neurotransmitter being released here. But you can see that the autonomic nervous system is going to trigger muscles, glands, and cardiac, specifically smooth muscles, glands, and cardiac muscle. And something that's a little different here is that the effect can be stimulatory or inhibitory, depending on the neurotransmitter, depending on the receptors that are actually on the effector organs. So we're going to talk about this term ganglion, but pay attention. If you remember, ganglion is just a collection of cell bodies. So then this is going to be the preganglionic neuron, and this is going to be the postganglionic neuron. The other thing to notice is the myelination. Where we had heavy myelination in the somatic nervous system, when we get to the autonomic, we're going to have lightly myelinated pre's. You'll notice that all the pre's have myelin. What about the posts? Unmyelinated, unmyelinated. So the posts are unmyelinated in the autonomic nervous system. I think this is a great question. What's the advantage to having this two neuron system instead of just one? And this is something you should be asking as you kind of move through anatomy, especially when you get to 202. You should be asking yourself why. There's a reason that it's more complex. What's the advantage to us by having this two neuron system? Well, one, it allows for neuronal convergence. So this is multiple pre's, so many pre's talking to one post. You can also have neuronal divergence, where this is one pre reaching out and talking to many posts. So you can see how this would overlap information, and this would allow for one signal to reach out and basically talk to several different posts that would all have cumulative effects. And I'll show you some examples as we move through the PowerPoint. We can see that there are some brain and some spinal cord control of these autonomic functions. So the hypothalamus is going to kind of link emotions and autonomic function. Think of being scared and your heart races faster, right? Brainstem is going to control some of the reflex actions. So think about if you smell food, all of a sudden your mouth starts watering. That's a gland, right? And that's being triggered by an autonomic nervous system reflex. And then in the spinal cord is where we're going to have some of these lower order urination, defecation reflex centers. If we wanted to look at functional differences, simply put, parasympathetic is rest and digest. You've just eaten a really large meal, you're trying to digest that meal, and usually, since it also has something to do with pupil control, most people say you just had a really large meal and you're sitting down to read something. Sympathetic division really simply would be fight or flight. So this is an emergency, you're running away from something, but you also are activating the sympathetic division when you exercise. So the anatomical differences between these two divisions, one is where they exit the spinal cord. So craniosacral would be parasympathetic, thoracic, lumbar would be sympathetic. In parasympathetic, we're going to see preganglionic axons that are long and posts that are short. So we have a really long pre and we have a short post. The ganglia are located in the organ or very close to the organ. With sympathetic, we have short pre's and long posts. So you have this short pre, really long post, and the ganglia are close to the spinal cord. If we look at the degree of response, it kind of lends itself to the length of the ganglionic axons that we just talked about. So parasympathetic activity is localized. Remember we said we had the long pre and we had the short post. And we said the ganglion lies very close or within the organ. So this is a very localized response. I'm telling just this organ to reduce activity. With sympathetic, this fight or flight, we had a short pre and then we had a really long post. So the pre, also if you remember, the ganglion lined up next to the spinal cord. And remember we said that some preganglionics can reach out and talk to multiple posts. So we get this mass activation. Think of all the things that have to be turned on or enhanced or sped up if you're working out or you're running from someone, right? You have to continue to send information to the heart to speed up, to your blood vessels to constrict, to make your blood pump faster. You might want to tell your kidneys, hey, stop worrying about urine. We're fighting for our lives here. We don't want to have to stop and pee while we're running. So there's a lot of kind of mass activation with sympathetic where parasympathetic is very localized. I think this is a nice overview of the parasympathetic pathways. One, you can see they're coming from the cranial region and the sacral region. You can see that in most cases, the pre's are very long and the posts are very short, and you can see that the synapse or the ganglion is happening at the organ level. A nice overview of the sympathetic pathways. You can see they're coming from the thoracic and the lumbar region. We can see the sympathetic ganglion that's running alongside the spinal cord. I've had students tell me this reminds them of a service road, right? So your spinal cord is the interstate, and that service road that runs the length of the interstate, that's the sympathetic ganglion. And this is kind of the hub where we can activate multiple organs or have that mass activation that we just talked about. You can see how these kind of serve as hubs, the ganglion serve as a hub. So you could have this sympathetic nerve coming out at T1 and it can travel through 3 different ganglia and it could trigger eyesight, it could trigger heart, it can trigger respiratory. So all of those you would need if you're trying to run away, right? So hopefully this makes sense. You can see that there are shorter pre's and much longer posts and that the ganglions are running right next to the spinal cord. When we're looking at the spinal nerve pathway for sympathetic here, and I know this is sympathetic because I can see the sympathetic ganglion chain here. If you remember, we talked about this little loop called Rami communicants. If I can squeeze all that in, there we go. So remember, we've got dorsal root, ventral root, merges to spinal nerve. Then we have posterior ramus, anterior ramus, and then we have this little loop here in the sympathetic ganglionic chain. And what's happening, you can see that the motor neuron is coming out through the ventral root, and it's looping through that communicant to the ganglion. It can also, if I'm on a different level here, this is just another motor neuron coming out, it can actually use the communicant to go up. So this is how we can have one pre that's basically talking to several post-ganglionic neurons. And again, when we're talking about this sympathetic, we're talking about kind of mass activation, getting everybody going in the same path with the same message. I just quickly wanted to point out that there is some continual branching that we'll see. There's things like the cardiac plexus, the pulmonary plexus, and in this branching, you're going to have both sympathetic and parasympathetic nerves. We have to mention the enteric nervous system. It's really pretty interesting. It is independent of the central nervous system. So there are autonomic motor neurons and visceral sensory neurons, but they're all embedded in the wall of the GI. They can talk to the central nervous system, but they're not controlled by the central nervous system. So they're going to coordinate peristalsis. So the smooth muscle coordinates peristalsis, which is alternating contractions to move food through your intestines. And we have glands that are producing mucus that line the small intestines to help facilitate the movement and the absorption of the nutrients from the food. So it's pretty interesting. Again, the enteric nervous system is independent of the central nervous system, but there can be some talk between these two divisions. Basically, if you were brain dead, you could still have your enteric nervous system working. If we wanted to compare neurotransmitters, first we would look at the parasympathetic, and we look at the pre. The pre releases acetylcholine. The post releases acetylcholine. And here we have our target cell. In sympathetic, we have acetylcholine is pre, acetylcholine is post, if we're talking about sweat glands. The pre is acetylcholine, the post is norepinephrine, if we're talking about some other body structure. Now, the medulla, adrenal medulla, is a little bit different. This is a gland that we won't really talk about until 202, but there are not two neurons here. This is one of the exceptions when we talk about sympathetic nervous system. So there's a preganglionic, and it also releases acetylcholine, and that's going to trigger the adrenal medulla to release hormones into the bloodstream. So what's something that you can tell right away here? All of the pre's release acetylcholine. Most of the posts release acetylcholine. Our exception is here. So let's talk about a specific example. We have our heart, or the cells of our heart. Your cells can have multiple receptors. Let's say this is acetylcholine receptor, and over here I have a norepinephrine receptor. So my parasympathetic releases acetylcholine. That acetylcholine binds the receptor, and it triggers the heart to slow down. Now, I'm a sympathetic neuron. I release norepinephrine. That norepinephrine goes to the norepinephrine receptor and binds. That triggers my heart to speed up. So most organs are dual innervated with both sympathetic and parasympathetic neurons, and these organs, these cells, have different receptors to either turn up or turn down activity. So of course it has to be more complicated. Now we get to talk about receptors. So we keep saying acetylcholine receptor, but there are different types of receptors, and that will cause different actions to happen. So you'll see that all of the post-ganglionic receptors are nicotinic receptors. And here we have nicotinic receptors on the adrenal medulla. Really, you can think of the adrenal medulla as behaving like a post-ganglionic neuron. So all the posts have nicotinic receptors. You can see that all parasympathetic have muscarinic receptors, and some of the sympathetic have muscarinic receptors. Lastly, we have to talk about this adrenergic. It's a very strange word. It's based on the word adrenaline. So we have norepinephrine and epinephrine that would both be hormones that we would call adrenaline. So we're talking about sympathetic pathway. We're talking about most other body structures. So here was sweat glands, here was adrenal medulla. So this is everything else. These are all of the other organs that we're going to speed up. So you're thinking about your heart, you're thinking about lungs, right? Blood vessels that you're going to constrict to have your blood pressure go up. So there's 5 different receptor types here. There's 2 different alphas and there's 3 different beta type receptors. So there's 5 different receptors and they all have different functions. Either they're going to be stimulatory or they're going to be inhibitory, depending on which organ they're on, which receptor. So there's a lot more play here than I want to get into, but I just want you to realize, why do we need to know about these different receptors and these different neurotransmitters? The reason is this is how we design drugs. So maybe you've heard of something called a beta blocker. A beta blocker comes in and blocks a beta receptor that you have on cardiac muscle. If your heart is beating too fast, we give you a beta blocker, and now everything slows down because you've blocked the receptor and the normal pathway, and so we have a slowing down. So we use these receptors to design drugs. And so I think it's important when you get to pharmacology to at least have some understanding of the different receptors, where they're located, and whether they're all going to be stimulatory. So acetylcholine is always going to be stimulatory, but that if you have epinephrine or norepinephrine, that these can cause inhibitory or stimulatory depending on the organ. Here's another good example in clinical treatment for asthma. So here we have asthma, right, which causes the bronchioles in your lungs to narrow. So you have decreased oxygen airflow. So if we give you epinephrine, it binds to your beta 2 receptors and it causes them to relax, and they dilate, and that increases oxygen and air. The same neurotransmitter on the heart, except for the heart has receptors that are beta 1, and when epinephrine binds the beta 1 receptors, it causes your heart rate to increase. So we have the same neurotransmitter doing both an excitatory event and an inhibitory event, depending on the organ and depending on the receptor. I kind of relate autonomic tone to muscle tone. So muscle tone was that partially contracted state. So certain organs have to maintain this partially constricted state. Some are going to be due just to one division, and some are going to be innervated by both divisions. So you could have autonomic tone regulated by the parasympathetic division or autonomic tone regulated by the sympathetic division. I mentioned dual innervation earlier. I want to make sure we understand that an organ receives input from both divisions. So if we're trying to control heart rate, we have both the parasympathetic, which can slow down the heart rate, and we have neurons from the sympathetic division that can speed up the heart rate. We also have autonomic reflexes. So these are going to happen in cardiac, smooth, or in glands. This is that pre-programmed response, just like we saw somatic reflexes. An important autonomic reflex is the cardiovascular reflex. This will make a little bit more sense when we get to 202, but there are stretch receptors in the blood vessel walls. So if you have a blood vessel that's in its normal a contracted tone, and all of a sudden there's tons of force being pressed on the wall from the inside. So if your blood pressure is high, well, this is not good for your heart. We don't want blood coming at really high levels back to the heart. It can damage cardiac tissue. So a signal is going to be sent to the brainstem. We can tell the sympathetic, right, inhibit the sympathetic, stop beating so fast to the heart. We can activate parasympathetic, that's the rest and digest, and that'll cause your heart rate to slow down, and that'll decrease the amount of blood that's being ejected, so you won't have this pressure of this blood inside the blood vessel. So ultimately it'll decrease your blood pressure. This is another good example of an autonomic reflex. As the urine collects in the bladder, the stretch receptors in the bladder will send a message to the spinal cord integration center, and that'll send a message out, motor message, for the muscles within the wall of the bladder to contract and for the sphincters to open so that we can release the urine. I think this is a great slide to go through. Look at the differences between somatic and autonomic. Tell me about the two different divisions. Tell me about the number of neurons. Tell me about the neurotransmitters, pre and post. Tell me about the organs that are being affected, and then whether we have stimulatory or inhibitory action. I'll talk to you guys next time. Bye.